You take that check. You take that. No, he's not able to check the check.
Hello, good morning. Hello, good morning. Hello, good morning. Hello, good morning.
Radiations ಬರುತ್ತಲ್ಲ ಕಾಲ್ ಬಂದಾಗ ಗುಯ್ ಅಂದ್ಬಿಡುತ್ತೆ.
Good morning.
Radiations ಬರುತ್ತಲ್ಲ ಕಾಲ್ ಬಂದಾಗ ಗುಯ್ ಅಂದ್ಬಿಡುತ್ತೆ.
Good morning.
Science knows no country because knowledge belongs to humanity and is the torch which illuminates the world. Good morning, esteemed guests, faculty members, and fellow enthusiasts of the fascinating world of science. I'm Sandana.
I'm Navya, and it's a great pleasure to welcome you all to this scientific event. Today, we are gathered here to celebrate the spirit of science and the pursuit of knowledge.
Science knows no
This
country.
event,
because
jointly organized by the
knowledge belongs to
Department of Plant
humanity
Pathology
and
and
is
the
the
Plant
torch which
Pathology
illuminates
Club,
the world.
promises to
Good
delve
morning,
into the
esteemed
fascinating
guests,
realm of
faculty
plant
members,
viruses.
and fellow enthusiasts of the
Today, as we
fascinating
commence
world
the
of
first
science.
lecture
I'm
series of
Sandana.
Plant Pathology Club, let us and it's a explore the great pleasure
intricate
to
world of
welcome you
Gemini
all to this
viruses
scientific
and also
event.
reflect on the
Today,
profound
we are
connections
gathered here
between
to celebrate
science,
the
plant
spirit
health,
of
and
science
human
and the
progress.
pursuit of knowledge.
Science
We're
knows
truly
no
honored
country.
to have some
Because
esteemed
knowledge
personalities
belongs to
join us today.
humanity and
Before we
is
proceed with the
the
program,
promises
I'd
to
like to
delve into
invite
the
our distinguished
fascinating
guest
realm of
to
plant
grace the
viruses and
dais.
fellow
So
enthusiasts
firstly,
today, as we
I'd like
commence
to
the
heartily
first lecture
invite
series
Dr.
of
B.
Plant
Muniappa
Pathology Club,
to this
let us
special
explore the
occasion, and
intricate
we would
world
like
of
to
Gemini
express our
viruses
deepest
and also
gratitude
reflect
for
on the
your and your
profound
family's
connections
presence here
between
to join us on
science,
this
plant
event.
health, and human
And the
progress.
pursuit of knowledge.
Science
We're
knows
truly
no
honored
country.
to have some
Jointly
esteemed
organized by the
personalities join us
Department of
today.
Plant Pathology and
Before
the
we
Plant
proceed with the
Pathology
program,
promises
I'd
to
like to
delve into
invite
the
our distinguished
fascinating
guest
realm of
to
plant
grace the
viruses and
dais.
fellow
So
enthusiasts
firstly,
today, as we
I'd like
commence
to
the
heartily
first lecture
invite
series of
Dr. B.
Plant
Muniappa
Pathology Club,
to this
let us
special
explore the
occasion, and we
intricate
would
world
like
of
to
Gemini
express our
viruses
deepest
and also
gratitude
reflect
for
on the
your and your
profound
family's
connections
presence here
between
to join us on
science,
this
plant
event.
health, and human
And the
progress.
pursuit of knowledge.
Science
We're
knows
truly
no
honored
country.
to have some
Jointly
esteemed
organized by the
personalities join
Department
us
of
today.
Plant Pathology and
Before
the
we
Plant
proceed with the
Pathology
program,
promises
I'd
to
like to
delve into
invite
the
our distinguished
fascinating realm
guest
of
to
plant
grace the
viruses and
dais.
fellow
So
enthusiasts
firstly,
today, as we
I'd like
commence
to
the
heartily
first lecture
invite
series of
Dr. B.
Plant
Muniappa
Pathology Club,
to this
let
special
us
occasion.
explore
It's
the
also our absolute
intricate world
privilege
of
to
Gemini
invite our
viruses
guest
and
speaker,
also reflect
Dr.
on
K.
the
Ganeshaya,
profound
to
connections
grace the
between
dais.
science, plant health,
And now I'm delighted to invite the Vice Chancellor of UAS Bangalore, Dr. S. Suresha. We invite you to join everyone.
Thank you all for gracing us with your presence. We are honored to have such distinguished individuals with us today. As we move forward with the program, I encourage all participants to engage actively and make the most of this enriching experience. Today's discourse inspire curiosity, ignite passion, and serve as a testament to the transformative power of science in shaping a better tomorrow. Thank you for joining us on this enlightening journey. Let the exploration begin. We are honored to have.
But before we begin our journey into the depths of knowledge, we have a few opening moments to set the tone. I would like to invite Kritika from second year PhD to the stage for a prayer song. Let's all join together in invoking positivity and inspiration for the proceedings ahead.
ನಮ್ಮಮ್ಮ ಶಾರದೆ ಉಮಾ ಮಹೇಶ್ವರಿ ನಿಮ್ಮೊಳಗಿಹನಾರಮ್ಮ ನಮ್ಮಮ್ಮ
Let's all join together in invoking positivity and inspiration for the proceedings ahead.
ಕಮ್ಮಗೋಲನ ವೈರಿಸುತನಾದ ಸೊಂಡಿಲ ನಮ್ಮಮ್ಮ ಶಾರದೆ ಉಮಾ ಮಹೇಶ್ವರನಾದ ಸೊಂಡಿಲೊಳಗಿಹನಾರಮ್ಮ ಗಣನಾಥನೇ ನಮ್ಮಮ್ಮ
Let's all join together in invoking positivity and inspiration for the proceedings ahead.
ಶ್ವರಿ ಕಮ್ಮಗೋಲನ ವೈರಿಸುತನಾದ ಸೊಂಡಿಲ ನಮ್ಮಮ್ಮ ಶಾರದೆ ಉಮಾ ಮಹೇಶ್ವರನಾದ ಸೊಂಡಿಲೊಳಗಿಹನಾರಮ್ಮ ಗಣನಾಥನೇ ನಮ್ಮಮ್ಮ
Let's all join together in invoking positivity and inspiration for the proceedings ahead.
Raju for his welcome speech. Welcome speech.
ಎಲ್ಲರಿಗೂ ನಮಸ್ಕಾರ. Good morning to all of you. Respected personages on the dais, respected member of board management, respected officers of the university, respected former HODs, senior professors, family members, students, and well-wishers of Dr. V. Muniappa sir, and various department students and friends, invitees from industries, institutes, and students from UHS Bangalore PG Center GKVK. So indeed, it's a great occasion that the Department of Plant Pathology is organizing a definite purpose. This program is organized with a definite purpose, namely Plant Pathology Club Lecture Series and honoring eminent personalities. In this regard, before welcoming the dignitaries, I wish to read out the brief profile of the Department of Plant Pathology. So Dr. Leslie C. Coleman in 1908 undertook a comprehensive studies on important diseases of crops of the state and tackled the problems which later marked the beginning of the plant pathology. The teaching of plant pathology was started in 1913 at Hebal Agriculture School, and full-fledged department came into existence in 1946 with the establishment of Agriculture College at Hebal. The postgraduate program in plant pathology was started with the launching of USB in GKVK during 1964. The department has led by eminent pathologists and personalities, namely Dr. H.C. Govindu, Dr. K. Ramakrishnan, Dr. K.G.S. Shetty, Dr. H.R. Reddy, Dr. V. Muniappa, Dr. K. Krishnappa, and Dr. E.N.A. Khan, and others. It is a great pleasure to mention that the Department of Plant Pathology has bagged 6 times the prestigious ICR Jawaharlal Nehru Award with a beginning in 1975 from Dr. V. Muniappa. That is an outstanding research in plant pathology. So it's a proud moment that Dr. V. Muniappa is with us. So he was the first person who received the ICR Jawaharlal Nehru Award. So the department has expertise in various disciplines, that is mycology, bacteriology, virology, nematology, which conduct both basic and advanced research. So totally 171 PhD and 519 MScs were graduated from the inception of the department. The contribution of the students, teachers with the awards and research publications and technologies from the department is commendable. The department is collaborated with the different organizations, so for funding both national and international level. The department is equipped with the Biocontrol Laboratory, the Seed and Plant Health Testing Laboratory, National Certification System for Tissue Culture Raised Plants, NABL accredited Plant Molecular Diagnostic Laboratory, which caters the needs of farmers and industries. The vision of the department is to strengthen the graduate students with skills in plant pathology, besides hands-on training and motivating postgraduate students to pursue quality research to cater crop protection needs of the farming community. The department also has envisioned to conduct various science-related activities by establishing the Plant Pathology Club. So today is the opening day of the Plant Pathology Club, and as the first activity of the club, it is wished to organize a guest lecture by eminent scientist by Dr. Ganeshaya under the Plant Pathology Club lecture series. So further, it was also thought to honor the plant pathologists who contributed immensely in the field of plant pathology in US Bangalore. On the occasion, and today, we wish to honor Professor V. Muniappa, the eminent virologist who served in the field of plant virology on his 80th birthday. Today is his 80th birthday, to make the program more memorable. So this is about the brief profile of the department. Now, in this regard, or during this occasion, so I feel myself proud, and it is an immense pleasure to welcome our beloved Honorable Vice Chancellor, Dr. S.V. Suresha sir, to grace the occasion for successful beginning of the Plant Pathology Club lecture series. So on behalf of the members of Plant Pathology Club and participants on my own behalf, I heartily welcome to this program, sir. I take this opportunity to welcome Dr. K.N. Ganesha, sir, INSA Emeritus Professor and my teacher, who is kind enough to accept our invitation to deliver a guest lecture. Sir, your presence has really brightened our program. So on behalf of the members of the Plant Pathology Club, on my own behalf, I heartily welcome to this program, sir. So it's a proud moment to welcome our beloved mentor, eminent personality, former professor and head department of plant pathology, Dr. V. Muniappa, to this program. Sir, on behalf of all the participants, and heartily welcome you to this program, sir. I take this opportunity to welcome the members of Board of Management, Dr. H.L. Harish, Dr. H.L. Harish, and Dr. Chandregoda, sir, for having accepted our invitation and graced this occasion. Thank you very much for your kind presence, sir. I take this opportunity to welcome our former registrar, Dr. Jairamaya, sir, former dean and also professor head and my mentor, Dr. K.V. Keshamurthy. So head division of plant protection, Dr. Krishna Reddy, former professor and head. And I also welcome the librarian, Mr. Dr. Pape Gowda. I also welcome Dr. R.S. Kulkarni, sir. I also welcome Dr. Sampangi Ramaya, sir, alumni of our department. Now he is presently in US. So I'm very thankful to you, sir, for having accepted my invitation, and your presence is greatly appreciated. Yeah. So I also welcome ADR Dr. Uma MS. I welcome the senior professor and emeritus professor, Dr. Viratmat, sir. I welcome all the heads of the department, former heads of the Department of Plant Pathology, faculty and students and friends to this program. I also welcome seed and members from the seed and pesticide industry for this program. I welcome the family members, particularly Madam Sudha Muniappa. Sudha Muniappa, relatives and friends, daughters Suti, Vinay, daughter-in-law, and then the grand, lovely daughter. I also welcome all the participants who have joined online. Many of them have joined online. So once again, I take this opportunity to welcome all of you, all the heads of the departments, students and friends, relatives, well-wishers of Dr. V. Muniappa, for having graced this occasion and made this program more successful. I wholeheartedly you one and all. Sarve Jana Sukhino Bhavantu. Thank you very much.
Thank you, sir, for extending your warm welcomes to everyone. The Plant Pathology Club, envisioned by the Department of Plant Pathology, is ready to unfurl its wings and soar into the realm of advancing scientific knowledge. It stands as the embodiment of a shared dream, eager to contribute to the expansive landscape of discovery in science. This initiative is not merely an organization, but a collective aspiration to delve into the intricacies of plant health, fostering a community
Very-
dedicated to learning, exploration, and collaboration.
And what more fitting moment
Conversation can
with the plant pathology? Now, I'd like to extend a heartfelt invitation to our esteemed Vice Chancellor, Dr. S.V. Suresha, to join us in inaugurating this significant initiative. And I also request him to unveil the logo that encapsulates the spirit of the Plant Pathology Club digitally. Please, sir.
આમ કરાય ઉપરથી ખેંચ છે એટલે આ બધું જતું રહ્યું જો અરે ઘોરે
I'd also like to invite the president of Plant Pathology Club, Dr. N.
Yeah,
Nagaraju,
that's-
the secretary, Dr. C. N. Lakshmi Narayana Reddy, and the treasurer, Dr. Manjunath B., to join us in the inauguration.
Thank you, sir, for officially inaugurating the Plant Pathology Club and unveiling its emblem digitally. Your presence on this special occasion adds a touch of distinction, and we eagerly anticipate your guidance as we embark on this exciting journey of knowledge, discovery, and growth. I'd also like to thank the core members of Plant Pathology Club for joining.
Now that the club is officially inaugurated, we would like to brief the objectives of the Plant Pathology Club.
The rationale behind creating this platform is to connect postgraduate and doctoral students of the department to its alumni working in different sectors and other stakeholders in the field of plant pathology. The network serves as a rich resource, fostering mentorship, sharing experiences, and opening up avenues for collaborations.
We'll be hosting a series of engaging events, including lecture series featuring expertise from academia and industry, providing insights into the latest developments in plant pathology. We'll also organize training programs and skill-oriented workshops aiming to equip students with practical knowledge and problem-solving abilities, aligning them with the evolving needs of the industries, which is essential for addressing real-world challenges. Beyond that, this club serves as a hub for sharing job openings, conferences, and valuable resources, creating a dynamic ecosystem that benefits student community.
We believe in honoring those who have paved the way in the field of plant pathology. A club will recognize and celebrate the achievements of individuals who have contributed significantly to this discipline, thus fostering a culture of motivation and inspiration among students by showcasing success stories and achievements within the field.
Moreover, our commitment extends to fostering collaborations and partnerships to strengthen our community and support each other's professional journeys. By uniting our expertise, we can make a significant impact on the field of plant pathology and sustainable agriculture.
With these objectives and aspirations, we wish the Plant Pathology Club a great start, and we hope for the active involvement of the enthusiasts of science.
Now, brace yourselves as we shift gears and move on to the main highlight of our program. We have an esteemed guest speaker who will be sharing some invaluable insights with us. But before that, I'd like to invite Chetan from second year PhD to introduce our distinguished guest, Dr. K.N. Ganeshaya, and to share with us a bit about his background and achievements. And I also request that the dignitaries on the dais to be seated in front of the stage, as we have a guest lecture. So I request the dignitaries to shift. Sir, it's okay. It's just a short introduction. So
I would
please.
rather now prefer the time to be given to me. Yeah.
So just 2 minutes.
No, I will introduce myself.
So that will be an encouragement to other students. as well.
ಮುನಿಯಪ್ಪ ಬಗ್ಗೆ ಹೇಳೋದು ಸಾಕು, ಹಾ? Yeah.
Just 2 minutes, so please. Yes, yes.
Really? ಇಲ್ಲ, ಬಹಳ ಇದೆ ಬೇಡ ಬೇಡ. ಸರಿ ಮೈಕ್ ಆಗ್ತಾ ಬಿಡಪ್ಪ. ಥ್ಯಾಂಕ್ ಯು ವೆರಿ ಮಚ್. ರಿಮೋಟ್ ಕೊಡ್ತೀರಪ್ಪ. ಒಂದು ನಿಮಿಷ. ಎರಡು ನಿಮಿಷ, ಎರಡು.
ಇದು ಅಲ್ಲಿ ನೆಟ್ ಬರುತ್ತಾ? ಬರುತ್ತಾ?
Can you hear me? There are two speakers here. Why? That means I'm giving 2 talks. If at all there is any honorarium, you have to give me twice the honorarium. Yeah, let me have the slides on the screen.
ಈಗ get up quickly.
Hello?
Connect
Can you hear
internet
me?
connect ಮಾಡಿ. There's a good talk.
ಇಲ್ಲ.
ಈಗ ಬನ್ನಿ, ಹೆಂಗ್ ಕನೆಕ್ಟ್ ಮಾಡೋದು?
ಬಟ್ ಬಹಳ ಹಿಂದೆ ಇದಾರಪ್ಪ.
ಕನೆಕ್ಟ್ ಆಗಿದೆಯಾ?
ಹಲೋ,
Zoom ಬರುತ್ತಾ?
ಕೇಳಿಸುತ್ತಾ?
ಬನ್ನಿ, ಹಾಕ್ ಬನ್ನಿ. ಈಗ Zoom ಲಿಂಕ್ ಹೆಂಗೆ ಬೇಕು?
ಹಲೋ, ಈಗ ಕೇಳಿಸುತ್ತಾ? Not through the mic. So can you hear me otherwise? Oh, great. Yeah. Can I have question, please? First,
Yes, we can hear you, sir.
Professor Nagaraj approached me with a request that I should speak on this particular occasion. I very happily accepted because of several reasons which I'll come to. And we are now paying our felicitation to Professor Muniyappa, the famous virologist. And I was also asked to give a technical talk. So I have now also brought a technical title for my talk. But what I'll be doing is a mix of both of them. In that sense, it won't be a purely technical talk, but I'll be first paying my respects to Professor Muniyappa. And how do I pay my respects to Professor Muniyappa? Obviously, recollecting my own first meeting with Professor Muniyappa. It was the year 1976, and I had just passed my BSc Agriculture, was entering into MSc Agriculture, and Muniyappa in our district, the entire Kolar district by then, was very well known. He was young. I think he was married in 1975. I'm correct, madam? Yeah. In 1975, we got married, and by 1976, better believe it, ado because barutte. ಆಮೇಲೆ ಮಾಡಿರಂತೆ ಡಿಸ್ಟರ್ಬ್ ಆಗುತ್ತೆ ಅದಕ್ಕೋಸ್ಕರ. ನನಗೆ I want to talk about Muniyappa, not to now show myself, please. He was very young then. ಇದು ಬರುತ್ತಾ? ಇದು ಯಾಕೆ ಎರಡು ಕೊಟ್ರಿ ಇಲ್ಲಿ? But he was very well known in the entire district as a great scientist. By then he had become an international scientist, a well-known international scientist. He had toured the world across, and he also had got this Jawaharlal Nehru Award, and about this award, the entire district was talking about. I know he has got several more awards, but from my side, from the Kolar district, Professor Muniappa is only known as Jawaharlal Nehru Awardee Muniappa. So obviously there was an awe about Professor Muniappa, and people used to talk with lot of awe around him. So as I told, I was entering my MSc Agriculture, and there were two alternatives for me: one to enter into seed technology and one to enter into plant breeding, genetics and plant breeding. I, of course, had decided to go to plant breeding and genetics, but my father thought seed technology is a new subject that may have a lot of opportunities, like any father would think about. He suggested that I should also consider seed technology. So what to do? We couldn't decide, and my father told, let us go to Muniappa. Muniappa might have forgotten. We went to him, and where did we go? We went into this Kashi of Muniappa, the Kailas of Muniappa. Any of you? This is the building where Muniappa was working, and as we enter in, you see that corridor, and at the end of that corridor, there was that door through which I entered. I wanted to revisit the place again. So last week I went. Unfortunately, the door is become almost dilapidated like that. And towards the end of that corridor, there was his greenhouse, famous greenhouse, where he had all his experiments done. So we went and we sat with him. My father told, my son can't decide. Incidentally, my father introduced himself. As I told, I am also from Kolar district. We are also distantly related. So my father wanted to open out with that link. I am this, I am that. Namgu nimgu ish sambandha ide. Nan magan karkomandidini. I don't know whether Muniappa understood all that or not, but he acted as if he understood. And I asked, so yen samachara? Avanige seed technology hobeka, plant breeding hobeka? He turned to me, Nimge yen ishta? I told genetics and plant breeding. Matte hogi serkolli. So the contact or discussion was brutally cut short by Professor Muniappa, and that was the end of it. My father came out. And so I thank Muniappa for retaining my interest in genetics and plant breeding. And ever since then, whenever I used to go to the bakery, which our vice chancellor knows very well, from the bakery side, his lab looks like this. I always used to thank Muniappa whenever I went there, because it is because of his decision that I went to genetics and plant breeding. Big clap for him for this reason. And today, that particular building is tender processing cell. There is a huge board on that particular building. At that time, we of course knew very little about the Gemini virus that Muniappa was working upon. We know that it belonged to the family Gemini viridae. It's a non-enveloped twin virus because it occurs always in pairs like Gemini zodiac. And it has a single-stranded DNA, sometimes one copy, sometimes two copies, with the length of 2.5 to 5.2 kilobases. It infects wide range of plants. That was our problem, the agricultural problem. And it is transmitted by white flies, leafhoppers, aphids. This is all that we knew about Gemini virus. Of course, later we were also told in the pathology course that there are 14 genera in Gemini virus. However, when we started learning about Muniappa, among ourselves, this is a secret, sir, don't get angry on me. We used to call Muniappa as a combo lunch plate. I'll tell you why. I don't know how many of you remember, during 1972, lunch or dinner in Karnataka means only rice and rasam and sambar. Agree? Only during 1980s, the chapati and roti began to move from north. When it started coming, some hotels began to provide a combo lunch, which provides both chapati, rice, palya, and other things. So that combo plate of lunch was very famous. And knowing the range of work that Muniappa was indulged in, our friends used to call him combo lunch scientist. That is because you will now see that Muniappa as a scientist was all in one. We all know that a scientist has no boundaries and he can work on anything once he reaches that stage. Once he works on the demands or the needs and opportunities available for him, and he wouldn't care for the boundaries, whether it's pathology, entomology, or plant breeding. And Muniappa was one such a scientist. Though he was a virologist, of course he worked on virology. He also worked on entomology. He also worked in plant breeding. He also contributed to molecular biology, and of recently, he also has served as a science manager in some of the companies. So he was an all-in-one scientist, and so we were calling him the combo lunch plate at that time. Regarding plant breeding, any of the plant breeders would be always proud to have one variety released. But with Dr. Kulkarni, Muniappa has released 3 varieties of tomato that are resistant to the tomato leaf curl virus disease, just not released. In fact, the entire South Indian farmers owe Professor Muniappa a lot because they could cultivate tomato today without any fear of the virus because of these 3 varieties. And thus, his contribution to farmers invaluable. Then I told he's also contributed to entomology. I wanted to really know about Professor Muniappa from his contemporaries or his teachers. So I met Virat Mutt very accidentally the other day and told him, sir, I've been given this responsibility of talking. He congratulated me first. Congratulations, you have got a very good honor to speak on Professor Muniappa. And then he started telling some of these statements. What did he say? Ganeshaya, Muniappa is more an entomologist than a pathologist. Why? Because he was maintaining cultures of 4 species of insects almost for 2 to 3 decades. Some of the cultures were even difficult to be maintained by the entomologists, whereas Muniappa was maintaining them. And then he also told, and he also told, okay, ನೋಡಿ ಅಲ್ಲೇನು ಮೂವ್ ಮಾಡಬೇಡಿ ಕರ್ಸರ್ನ ಅಲ್ಲ ತೆಗೆದುಬಿಡಿ ಅದನ್ನ ಆರಾಮಾರ ತೆಗೆದುಬಿಡಿ yeah. He also told, in a time or in an era when molecular techniques had not even evolved or developed, he studied the viral strains purely using the process of serology. So that was, in fact, his efforts that actually yielded it. He also told me, Professor Muniappa identified specific biotypes of Bemisia tabaci for the first time, and also showed that some of those strains only can transfer tobacco leaf curl virus more effectively than others. And Professor Virat Mutt was very proud to say this. He told, I am proud to say that I worked with him on sandal spike. Muniappa and Virat Mutt toured the entire Karnataka state and South India for surveying the sandal spike. And what is Virat Mutt also contributed to him was he developed his own survey method and sampling technique where there was nothing existing to survey this sandal spike. Finally, Virat Mutt also put a complaint on Muniappa. What is the complaint? He told he was highly dedicated, hard-working scientist, used to be in the lab from morning to very late in the night. Now, I knew Muniappa is a hard worker, but I wanted to find out how late he was working. So I did a small sting operation. The sting operation, as I went to his lab, took the photo, and then called his wife. His wife narrated to me a very interesting story. Now, the reason why I showed this photo is this whole place was snake infested. Now, as I told, they got married in 1975, and by 1977, could you kindly stand up? They had their first son. He was 11 months old by then. She is alone. She is from a very comfortable family. Muniappa has put her in D quarters, an isolated remote corner of the campus. All around that, there are these casuarina trees producing wooling sound that that creates a kind of weariness in you. She's alone. And the fear of isolation and this eerie sound is making her feel very, when a mother is taking care of the baby, she is also psychologically very, very stressed. So every day she waits for Professor Muniappa, and that day also she waited, not the day, night. Most of the professors generally go back at 4 o'clock, not Muniappa. 6 o'clock is not there. 7 o'clock is not back home. 8, 9 is not back home. Alone at home with her first son. 11 o'clock, he has not turned up, and she already knows that the whole place is snake infested. 1 o'clock, he has not come back. Can any of you imagine what would be her position? So she decided, I have to now make sure what has happened to him. Snake infested area, 1 o'clock in the night, he has not come back. A normal person would generally at least rush back home to see the first son who is 11 months old. He has not come back. So what has happened to him? Breaking all her difficulties, she woke up the neighbor, Dr. Vishweshwara Gowda. Many of you know him. Vishweshwara Gowda being a very nice gentleman, he took her with the kid and went to the lab. And she was expecting that Muniappa will show a kind of appreciation for the concern that the wife has shown. But was that not to be? He told, "Yen bandiddo? Yenagide nange?" He outrightly dismissed her concern. Well, his stories of late night and all this labor was also confirmed to me by his daughter, Shruti. When I called her, she told, "Uncle, he used to take me to the laboratory saying only 1 hour gamma obidana." But he used to now engage me for the entire morning and afternoon. A small kid of the kind of Aditi, she would like to play with the kids, with Jai and others. But he used to now take her away to the lab, spend the entire morning or evening, and kill her morning and afternoon. Professor Muniappa, you have been very, 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 However, Shruti, where are you? Please stand up. That is his daughter. I'll tell you, you should be happy. The reason why I say you should be happy is, I scanned a lot of photographs of your father. Generally, he's tightly put like this. Even when he was photographing here, when he was welcomed, he hardly opened his mouth. That is Professor Muniappa. He never shows his teeth. But one thing I found, whenever he is with his daughter, he shows his frontal teeth. Believe me, Shruti, I saw all the photographs. The other other addition after you is, he opens his mouth much, much more when he is with his granddaughter and daughter. And I just now spoke to his granddaughter. She told, "Yes, no wonder, uncle, or tata andamya gotillange." She told, "My grandfather is at my command, and not that I am at his command. I used to show whatever I want and he used to get me." Muniappa, congratulations, for at least once you have yielded to someone. Otherwise, Muniappa won't open his mouth, you see. Even in front of the foreign scientists also tight-lipped. And he has another style. Whenever he's with some foreign scientists, his confidence, he used to put his left hand like this and stare at the foreign scientists, as you can see there. So that is Professor Muniappa. And I spoke to a lot of his friends and colleagues and gathered a kind of personality analysis of Professor Muniappa. And I can describe his personality with the 3 A's. The first 2 A's are, is an admirable person, an adorable person, because his students adored him, his friends admired him. Of course, world over, his colleagues respect him a lot. His family loves him. His family loves him. University obviously was very happy because he contributed 3 varieties and also some control methods for tobacco leaf virus. But in contrast to these 2 A's, there is a 3rd A, and that is, he was rather alienated, rather away from everyone, never used to mix with anyone. And this was a contradiction. His students were always afraid of him. So was Shruti. Whenever Shruti used to go to the lab, it seems not only he spoils her morning and afternoon, but he used to know order. Correct, Shruti? So that is the kind of discipline he had. And so his students were afraid of him. Colleagues also were afraid to approach him. His family and relatives always wanted more of him. I don't think he has given enough to their family, though of course they don't complain on that. And university never enlisted Muniappa for any of the, committees or meetings. He was not in any. And that is not liked by many. Many won't digest. He will now tell on the face whether it is vice chancellor or dean. no, this is not correct. And so they didn't want Muniappa. They wanted always an Ejiram. Nevertheless, his students love him so much that this is a photo of 1970, his 75th birthday, and they always want to celebrate. Look at the hall. So many people have gathered here, and that is the kind of culture that Muniappa has left behind, and that is the kind of love he has left behind with the students. Though they are afraid, they all love him, so they come back to him. I also found one thing. I did say you won't laugh, but look at this. Padmaja, where is Padmaja? She gave me this photo, and I was told the other lady is Anita. Yeah, she's there. And look at it. He has always pushed his students to the front. He's standing with the foreigner there. He always used to now promote his students, and that is his greatness. I went to his lab. I recently told the lab that you photograph there is something like that. But Padmaja, I traced that particular lab. The frames are still there in that place. If any of you want to know how some fossil and some relic and some memory of your Muniappa's lab, the frames are still there because perhaps no one wants them. The whole lab is dismantled. Look at the frame shape. They're exactly the same. The frames of the greenhouse in front of which he was standing is still there. I think a small piece of it can be brought and kept in the Department of Pathology. Well, in addition to that, I also came to know that Muniappa loves several things. One among them, of course, is Gemini virus. I heard some complaints from some students. What is that? 2 days they would have surveyed. They're coming on the last day. They thought the work is over. Car is moving. They're all sleeping. Muniappa would say, stop, stop. Why? You saw some virus plant there. Even despite hard work for 3 days, you won't be satisfied if he now sees a new virus plant. And in addition to that, he almost always liked his snake-infested greenhouse and the lab. There is another important thing that he loved, his typewriter. He loved his typewriter because at that time, as you know, there were no computers and he had to always now send papers to outside. He has to type on his own. You wouldn't believe in the typewriters. The errors are abundant. So he used to sit and type when 10 o'clock, 11 o'clock, 12 o'clock, 1 o'clock in the night. Newly married wife would always want his company in the night. No, he's not to be. He will sit in front of the typewriter and type. I thought Sudha would have destroyed this typewriter after some time. Apparently not. She also has love for this typewriter because she loves Muniappa and Muniappa's work so much that is still intact. And I got the photo of that. He has other things that he loves. The camera. It is, look at the shape in which this camera is. It's still perfect. It is still working and so perfect. Thanks to Shruti for sending all these photographs. And one secret I'll tell you. You know, people at that time, the professor used to say, I bought the first car in my department. I bought that in my department. Professor Muniappa brought the first internet for the entire university to his house. But sometimes that internet never used to work. And so he used to now tell his daughter, a morning coffee coffee house. She used to happily join in, but he was actually cheating her. You know why? He was going to coffee house to work on the internet there in the coffee house. So, I mean, Shruti, he didn't know this. I got this message. And in addition to that, one thing I was told is that he dresses meticulously. He never comes out of his room in the morning unless he dresses very meticulously. In addition to that, I was told he used to get a lot of gifts from abroad to share them with his relatives. And obviously, all his relatives were very happy about the fact that he used to get his gifts from abroad. I'm sorry I can't show too many of the photographs of his family, but nevertheless, let me move on to his work. This is about his family, his personality. Professor Nagaraj had given me a big bundle of papers of all the work that Professor Muniappa has worked on. And so I took all those titles of those papers and prepared a frame of what is that Muniappa's contribution is. Now, each of these terms suggests what is his contribution, and size of that represents how much he has worked on, depending on the papers he has published. Yellow mosaic virus, host range, Gemini virus, phylogenetics, indigenous population, range of things. And if at all you want to now give any gift to Professor Muniappa, maybe you can take this frame and put in the back or in the front of it and then present it to him. That summarizes the whole range of what that Professor Muniappa has done. Botanical pesticides, tomato varieties resistant to monopartite, indigenous population, socio-economic impact also he has now worked on. So look at the range of the subjects in which he has worked on. Coming back to his snake-infested lab, whenever I look at this photo, I remember a hermitage of an ashram of a rishi or a muni. In some sense, to me, Professor Muniappa is almost like a muni. His name also has in the first part. Because he dedicated his entire life to a field that he now loved. He has over 200 papers or around 200 papers in diverse areas, released resistant varieties, developed control methods and mitigation strategies for the virus, and he never went after any positions in the university. If there is anyone who has now achieved so much, as much as Muniappa, they would first do is to apply an application for the vice chancellor position. Muniappa never chased any of those positions. That is because, as I told, he is a Muni. And he built a large team of students, inspired work all over the world, not only in India, but all over the world, helped several international organizations in managing their science, and is still working. Evidence? Look at this. People say he's retired, but I don't think he's retired. Even in 2000, after his retirement, 2003, 2012, Nagaraj told there is a paper in 2013 also. 70 years aged, Muniappa also is still working. I wish all of us get so much of strength to now work at that age, if at all, if it is possible. So in other words, Muniappa is really a Muni, and that extra yappa is simply added as a respect to him. And so as a result, I have something to suggest. If only there is a person like Muniappa who has contributed to the field as much in a Harvard University or in a Cornell University or in any university abroad, they would have perhaps now renamed this Gemini virus itself by a different name after Muniappa called Gemuni virus. And I would now urge here, at least for our sake, I think we should now rechristen Gemini virus as Gemuni virus, and I think all of you would agree with me. Now, let us move on to the other part of the technical part of this talk. What I thought was, it's perhaps not good for me to simply give something on the virus or virology. I thought, let me now start tracing Muniappa's contribution to the field of virology. So can we now see how much Muniappa's work or Professor Muniappa's work has actually laid foundation for some of the most recent achievements in virology? And that's what I did. So how Professor Muniappa's work has laid foundation for new developments in virology. For that, for some of you who don't know Muniappa's work and the basic skeleton of Muniappa's work, let us start with the very basics. What is that? There is tomato or any such plant infested by the virus, and you get the curly leaves, and on the curly leaves, these Bemisia tabaci or any such vectors such as aphids or whiteflies sit on. They now start feeding on. When they start feeding on, what happens? The virus gets into their stomach, as indicated by the red particles there. And when this virus-infested whitefly goes on to a normal tomato, which is healthy, it now sits on that and starts sucking the sap from the healthy tomato. At that time, in the process of sucking the sap from the leaf, it also ingests back those viral particles into the leaf. And so that also becomes the virus infected. That is a cycle. In the whole of this virus whitefly or virus vector cycle, the most important element is also this vector. In case of tomato leaf curl virus, it is Bemisia tabaci. Now, as I told, as Rick but also told, Muniappa identified that there are biotypes in Bemisia tabaci, and some of the biotypes are more effective in transferring the disease than other biotypes. So this is Muniappa's work. How has it contributed to the further work? I called some professors in pathology and asked the following question. Why is that there is difference among these biotypes in transferring the disease? The answer I got was, sir, that is the genetic difference among the whiteflies. But I wasn't happy and wanted to see if Muniappach's work has in fact led to some new developments. And there I found that, of course, let me skip this. I found that there is a paper in Science, Nature Scientific Reports very recently as I think 2013, which shows that there is a specific reason for the differences among the biotypes to spread this particular disease. What is that? For that, I need to now take you to China. In China, in 1930, this tobacco leaf curl virus was introduced. As early as 1930, it was introduced to China, but it never became a serious problem. It became a serious problem only in 2003. So people start analyzing why in 2003 this disease became severe. They found that it is because of a particular biotype of the vector that is there or that was introduced during 2003. In fact, in China, there are two types, B type and Q type. And among the two, Q type was introduced in 2003. And after the introduction of Q type in 2003, in China, the disease became very severe. So now they have a reason, or they have a base to question what actually causes this Q type to now increase the efficiency of disease. So they took this Q type and they analyzed the reasons for its effectiveness in transferring the disease. And they found that there is a fourth culprit, just not the virus, just not the plant, and just not the vector. There is a fourth culprit that is responsible for increased efficiency in transferring the disease. What is the fourth culprit? The endosymbiont. Now, as you all know, most of the insects have endosymbionts, and there are two kinds of endosymbionts, the primary symbiont and the secondary symbionts. Whiteflies have both primary symbionts and secondary symbionts. What is the difference between primary and secondary? Primary symbiont is an obligate symbiont. The insect cannot live without that primary symbiont bacteria, and the bacteria will not survive without the insect. And that is a primary symbiont. They are tightly linked. Whereas secondary symbionts are not obligatory, they are facultative. They can be now released from the insect. For example, Proteera is a primary symbiont and Hamiltonella is a secondary symbiont on the Q-type whiteflies. Remember, Q-type is the one that is actually transferring. They examined, and incidentally, the primary symbionts help the vector in nutrition, physiology, and in its own survival. Whereas the secondary symbionts do not have any such role, they only help in the evolution of that organism. The first one, primary symbionts, are difficult to cure because if the primary symbiont is removed, insect is dead. Whereas secondary symbiont is easy to remove. What do you do for that? Simply treat with the antibiotic, you can remove the secondary symbiont. So Q type has Hamiltonella, whereas the B type doesn't have Hamiltonella. So they started thinking, if this Hamiltonella is the reason why Q type has become more effective in transferring the disease. So what they did was, they wanted to test the role of this Hamiltonella. So they treated the Q type with the rifampicin antibiotic. As a result, the Hamiltonella could be removed from the Q type whitefly. So they have two stocks now. One is the original Hamiltonella plus that contains the secondary symbiont, and the second is Hamiltonella minus, which does not have Hamiltonella. They tested the efficiency of these two in transferring. Remember, the biotype is same. The only difference is one stock has the symbiont, another doesn't have the symbiont. They tested the effectiveness of these two. Remember, Q type is as such effective, but they have created two stocks. One is plus H and another is H minus. They tested, incidentally, as I told, because the primary symbiont cannot be cured at all, despite treating with the antibiotic, the Proteera has not been removed. Only the Hamiltonella is removed. How much? In the H minus, it is completely removed, whereas in the H plus, it was 3,600 times more bacteria infested, the Hamiltonella infested. So they tested what is the efficiency of this H plus and H minus in transferring the disease. They tested in three parameters. I think the pathologists know here very clearly about these three parameters. One is how much time the biotype or the stock takes in accessing the virus when it is feeding on the plant, and if once it is taken in, how long the virus is retained in the stomach, and third, what is the efficiency of transmission. Let me give you the data. Now, this is the data of H plus and H minus relative amount of virus in the body of the stock with the time being hours. So what they have done, they have left these two stocks on the virus infected plant, and after every 1 hour, 2 hour, 3 hour, they have sampled them out and tested how much of virus load is there in the body of that white fly. As you can see, there are two interesting results. One is H-type white flies take up viral particles much faster. For example, at 12 hours itself, H-type has around 5 units, whereas at 12 hours, it only has around 2.5 units. So H minus type takes less viral stock in a given amount of time. And then look at the absolute amount of viral particles taken by H plus. It is very high, whereas H minus takes very little of the viral particles. So in other words, the H type, which contains the symbiont, takes the viral particles fast compared to H minus, which doesn't have that symbiont. And the load is very less also in the H minus. Then look at the retention capacity. So the white fly has taken the virus. How long it is going to stay in the living form or in the regular form in the body of the white fly? So they tested this between the two types, H plus and H minus. As you can see, in H minus, it drops faster than the H plus. They calculated what is the rate of removal or rate of destruction or decrease in the load of the virus between H plus and H minus. You can see in H minus, the rate of removal is very, very less. The virus is not destroyed at all. Whereas in H minus, the rate of removal is very high. Means what? Somehow in the body of the H minus, when the symbiont is not there, virus is getting destroyed faster, which means that symbiont, Hamiltonella, is protecting the virus in the body of the fly. And that's why perhaps the Q type has become very effective in transferring the disease. So Hamiltonella seems to protect the virus in the fly. Then they tested actually the rate of transmittance. They now left 1 fly, 5 flies, and 10 flies of both H plus and H minus. And you see that just with 1 fly of H plus, the transmission was 60%, and with H minus 0. Whereas with the 5 and 10 flies, the Hamiltonella plus, that is the Q type with the symbiont, transfers the disease almost 100% efficiency, whereas in the H minus, it is as low as 20%. In other words, it turns out that even at low densities, H plus white flies transmit virus more effectively, whereas H minus does not transmit as effectively. And what is the difference? Only the symbiont. The organism is same. Thus, the difference in biotypes is because of the third party, that is a secondary symbiont, and the symbiont virus is actually being protected, is protecting the symbiont, bacteria is protecting the virus. In other words, the biotype differences are because of the secondary symbionts and not the genetic quality of the symbiont as such. It is possible to now transfer these symbionts onto the B type also, and then the B type also becomes very effective. So I will now perhaps stop here. I had brought 3 elements to now present, but I'm sure the time is running out, and so I'll skip the second one about the horizontal gene transfer because of viruses. I will go to the last or the third one. The third one being viral genomes and human evolution. We all know that COVID affected us, and when the COVID affected us, we all observed one thing very interesting. What is that? The vaccine was first given to the old people and adults and not to the kids. We all know that. The reason is, the belief or the reason is that there was a strong evidence to show that the kids are not going to be affected by the COVID virus as much as the adults. The question is why? Why is that the kids are going to be so less affected compared to adults? So old people and adults were vaccinated first. And the reason is old people are more susceptible to COVID compared to young. And the implication is that young are relatively resistant. Why should young be relatively resistant and old should be susceptible? In fact, the medical science also shows or has demonstrated that fetus in the womb of the mother is completely resistant to most of the viruses, almost 100% of the viruses. It's completely resistant. Not only COVID, any such virus. and they are very well protected against many of the viruses. Why is that? Now, all of you know that when a virus particles has to affect its host, what actually happens is there is this external envelope proteins, which are actually the recognizing elements of the host. So let us say that is the host, the human cell, and that is the structure of that particular envelope protein. That structure of, or the protein structure of that envelope protein goes and exactly fits into the human cell. And so the human cell is recognized, and then the viral particle gets into, the DNA gets into, and the replication happens, and the oral human cell is destroyed. We all know this story. In other words, infection requires that the receptors on the human cell are available, and they are not available. If they are not available, viral particle cannot infect at all, because if the receptor cell is not available, the protein cannot go and bind, so infection does not happen. Now, what happens in the fetus is that the whole fetus is protected by the placenta. And they found that this placenta is the first to be infected. If at all any infection has to happen, the infection has to first infest the placenta, and then it has to go into the fetus. The placenta has to have the receptors so that the virus can identify and infect. But what is surprising is that placenta does not have the receptors. Why it does not have the receptors? Not that the placental cells do not have receptors. They have receptors, but all of them are occupied. Occupied by what? By the viral protein. Where is this viral protein coming from? The viral protein is coming from the baby and the mother. How can the baby and the mother have viral proteins? They have the viral proteins produced either by the baby or by the mother because the genome of the mother and the genome of the human beings, and hence the genome of the fetus, has the viral genomes incorporated into us. How does that happen? Let me now go here. Humans are exposed to viruses for millions of years, a range of viruses. And whenever such infection happens, those viruses have left behind a small segment of their DNA, and those DNA segments are incorporated into our genome. And they're there in the fetus, and they're there in the mother. So when the fetus is growing, those genes are activated and they produce the protein. That protein goes and covers all the receptor cells of the placenta, making it unavailable for the viral particles to be recognized. So these viral fragments are incorporated into the human genome, and these fossil DNA segments are activated in the placenta. And so the viral proteins are synthesized and the receptors are occupied. Receptors of the placenta is occupied. And hence, placenta and the fetus is immune to be infected by the viral particles. In other words, what was Ravana, or an enemy for all of us, itself has become a friend for all of us, because it is broken into pieces and is now become a friend within our whole genome. There is evidence for that. Just a small piece of data. There is this paper in Science in 2022, which shows, or which is under the title Evolution and Antiviral Activity of Human Protein of Retroviral Origin. Precisely the COVID-like virus they have tested. What is the evidence? There are two kinds of evidence. Number one, DNA sequences of retroviruses are abundantly available in the human genome. They have screened the human genome and they've shown that there are abundant presence of these retrovirus genomes. And some of them code for retrovirus envelope proteins, but the functional significance of that was not known. And one particular gene, or supin protein, was found to be protecting the placenta. And this supin protein is produced by the viral segment in our genome. When they removed the supin protein from the placenta, virus could infect. And whenever the supin protein was added, they cannot infect. In other words, the supin protein, which is produced by the DNA segment, which actually is a viral segment, is protecting all our kids. So we're all sitting here thanks to those viral genomes present in us. Otherwise, the entire human population would have been wiped out a long time back. And that is one of the most recent data that is available. I thought I share that with all of you, that to celebrate the kind of exciting work that Muniyappa has done throughout his life. So the viral fossil DNA has helped our survival in the history. With this, I end my talk, and I take pleasure in paying my own respect to Professor Muniyappa, and please join me with a standing ovation to Professor Muniyappa. Okay. Thank you very much.
Let's take a moment to express our gratitude to Dr. K. Ganesha for presenting the amazing personality of Dr. Vishnu in the best way possible. Thank you, sir, for taking the time to enlighten us with your insights and knowledge. We truly appreciate the depth and clarity with which you explore the intricate relationships between Gemini viruses, plant health, and human evolution. Thank you, sir. I'm sure many of you have questions after such an informative lecture, but as we are running out of the time, I just request 2 or 3 persons with very important questions to engage in the discussion.
These are all things. Question is, why is that those viral genomes are there in our genome? These are all random, in fact, I missed one particular presentation in between. On plants also it is happening. The same viral particles are carrying the segments of DNA from one plant, let us say from tobacco, to another plant, let us say cotton, when the virus comes from one host to another host. So the virus has wide range of hosts. So when it jumps from one plant to another, the genes get transferred. Plant virus can transfer only through pollen grains, but pathologists can transfer to the viral particles. Similarly, in human beings also purely by accident, some genes, some segments of DNA get broken and incorporated. Yeah. In case of plant, there is part of the plant, entering the plants. In plant virus. Yes. Part of that episomal. Yes. In fact, that part I left out. There was, I told you, speaker size. So I wanted to move from there to transfer of genes in plants to transfer of genes in humans. But speaker. Any other questions? Yeah. Thank you for I just want to know that viruses keep on changing. They keep on mutation, keep on changing. Whereas in this, whatever human body or animal, but still they are intact. Yes, very interesting question. But you know that in any organism in evolution, there are 2 sets of genes. One what are called conserved genes, another are variable genes. Because these genes are very useful to us, they are conserved. Though they belong to the virus, they are there in our body, but they are going to be useful in the placental and baby production. And so they are conserved in our body. In fact, one can ask the other question. If they are protecting the baby, why can't they protect us? These are costly. To produce those proteins is very costly. If every one of us start producing all those proteins, we may not even become very healthy. Whereas at the baby level, survival is very critical. And all the resources coming anywhere from the mother. So the baby can produce 3 proteins at the cost of the mother's milk. Yeah. What about the synthetic unfortunately the virus with infect the mother? And how can we protect the baby because husband has sorry, I didn't get your question. What about the synthetic viruses that they create? Yeah. Unfortunately, COVID-19 or whatever it is. There are no synthetic viruses. There are only small segments of virus. Yeah. If suppose new virus is there. Possible. I mean, they can be incorporated, but there won't be, as I told you, this paper in 2022 is opening entirely new field. And so people are now also raising the question, how much of the DNA segment that we use as vaccine should be allowed? Otherwise, it might not get into. If it turns into a Rama, it's good. If it still continues to be Rama, it's very bad for us. And so there are these discussions now going. This is a very, very new area that I presented. 2022 is a year. Yeah. Oh, very interesting. Even the mosquitoes are 1 minute. There are two symbionts. One is the primary and secondary. Primary symbiont is known to help the organism to reproduce and to multiply. Secondary symbionts are not known. As I know. Whereas the secondary symbiont is helping the virus.
So there is the one symbiont that is Wolbachia that they have that is present in the mosquitoes. So that can be cured also. We can remove it also. That's yes. Thank you so much.
I have a practical question. Cultivate papaya, since we are in the British part of the when you cultivate, particularly right with water, when it goes to the commercial area, definitely the virus will scan. When it comes to the isolated land, as a kitchen garden, not separated, the department, it is fully totally restricted. If there is a mechanism of transferring this, no, I mean, it's not resistant, it is just that it is escaping. You know, there is a uniform plantation, there is more possibility of spreading from the virus and all that, which is a very, common thing of gene to gene hypothesis story, I think, transmitters know here. So when the population of the host is very abundant, there is more opportunity for all these diseases to spread. And so they get infested, not accidentally they might, but more often they are isolated and so they don't get infection. That's all. They are not resistant. I stop here. Thank you.
Once again, thank you, sir, for your valuable time and for sharing your expertise with us. Your lecture has undoubtedly enriched our understanding of Gemini viruses. Let's give him another round of applause. As we continue with the program, today, we are also celebrating a truly special occasion. We have gathered here not only to honor Dr. V. Muniappa on the day of his birthday, but also to take a profound journey into his remarkable life and achievements. He's here with us today, along with his close family members. Your presence adds warmth and significance to this gathering, and we are truly grateful to each and every one of you who has taken the time to share in the moment of this occasion with us.
As we delve into the heart of a celebration, we have a special treat for you. In a few moments, we'll be premiering a documentary that pays tribute to the life, legacy, and extraordinary contributions of Dr. V. Muniappa, whom we lovingly call as Virology Muniappa. So without any delay, let us now turn our attention to the screen as we showcase the incredible life and work of Dr. V. Muniappa.
Welcome to everyone gathered here to have a glimpse of an unsung Dr. Muniappa's journey, who made immense contribution to the growth of plant pathology in general and plant virology in particular, which had great impact in plant pathology. Before getting into his contribution, it is just on us to have a look into some of the earliest developments in the field of plant virology in the world. All of us knew the economic imposition made by plant viruses through their devastating effect on crop production worldwide, but the percentage of losses reported up to 100% in several crops threatening the food security of the world. The earliest literature available with respect to plant viruses mentioned about the positive side of them by adding aesthetic beauty to the infected plants. The poem written by Japanese Empress in 1752 AD praises the beauty of equatorial plants represented with yellow mosaic is a testament to that. Further, in the late 16th century, the pride of having tulip birds capable of producing colored straight flowers went on craze and fetched huge price in market, which resulted in unusual social behavior of tulip trade in Netherlands, which famously known as tulipomania. Until Adolf Meyer, Ivanovsky, and Benjamin experimented results on tobacco mosaic disease in the late 19th century, no one was having awareness about virus affecting plants. Then experiments of sap transmission passing slide through Chamberlain filter to rule out the bacterial role in the vessels to naming the virus as a contagion vivum fluid were important milestones in the history of world plant virology development. Some of the very important milestones we need to remember are crystallization of PMP during 1937 by Stanley and deciphering the composition of plant viruses as a nucleoprotein by F.C. Bowden and N.W. Creddy. With this background, now let us have glimpses of one of the finest plant pathologists of India's scene, Professor Muniappa, who contributed immensely towards study of plant viruses and phytoplasma. He is with us today on the occasion of his 80th birthday. We are honored with your presence and we are privileged to have you here. His journey from village to successfully international acclaimed scientist is a fairy tale and inspiring. This unsung hero of country made silent revolution in the field of plant pathology in general and plant virology in particular. His commitment to the work is not less than tapas by any sage we have heard from our mythological stories. Professor Muniappa was born into a farming family to the couple Srimati Muni Venkataraman and Sri Venkataramanappa on December 15, 1943 in the village Ikrasanahalli located in Kolar Taluk and district of Karnataka. He did his primary schooling at Ikrasanahalli, middle schooling at Kodur and high schooling and PUC at Kolar. After this, he completed his graduation in 1968, post-graduation in 1970 in agriculture and doctoral degree in plant pathology in 1975 at College of Agriculture, University of Agriculture Sciences, Bangalore. Later, the place in which he studied has become his karma bhumi for the rest of his life to unleash himself as a committed scientist. In the process, he emerged as the finest plant virologist of country. Anything, if we say about Professor Muniappa, will be incomplete without mentioning the names of Dr. K. Ramakrishnan and Dr. H.C. Govindu, who laid a strong foundation at Department of Plant Pathology, UAS, Bangalore, to pursue research on their prevailing problems related to plant virus and phytoplasma diseases in the Karnataka state. Their guidance inspired and motivated Professor Muniappa to pursue his early research career to study on phytoplasma and plant viral diseases. His initial research work started as PhD scholar under the guidance of Professor Ramakrishnan, working on studies on yellow dwarf disease in rice production in Karnataka state caused by phytoplasma, for which he was awarded with prestigious Jawaharlal Nehru Award for postgraduate research from Indian Council of Agriculture Research, Government of India in 1975. This was the first Jawaharlal Nehru Award to the credit of UAS Bangalore. His research career in the faculty position started during the year 1974-75 as an instructor in microbiology and then went on to the positions such as junior virologist, associate professor, and professor and head. After retirement, he worked as an emeritus professor for a short stint. His research works majorly focused on the Wi-Fi transmitted plant viral diseases. In addition, a significant work done on other viral diseases of crop plants as well. To give few examples of his works, studies on phytoplasma diseases, purification and characterization of Gemuni viruses infecting crop plants, establishment of epidemiological and serological relationship of Gemini virus infecting different host plants, development of serological and molecular techniques for identification of plant viruses, diversity and interaction studies on host Gemini viruses and whitefly Venetia tabacum. The most impactful outcome of his research work in collaboration with AVRDC, Taiwan, was development of 3 tomato varieties, Nandi, Sankranti, and Viber resistant to leaf curl diseases, which is considered as a major problem in tomato production throughout the world. The resistant source identified by him in these varieties was transpressed into many commercial hybrids developed by 17 seed companies to counter leaf curl disease, which resulted in huge economic gain among the tomato growers of the country. During his career as a scientist, he guided 21 MSc students and 19 PhD students. In the process, he created very good human resource in the field of virology. The students, guided and inspired by him, have spread across the world, occupying pivotal positions in research organizations, universities, and industries pursuing research in plant virology as the torch bearers of his legacy. He had a rich international exposure in his early research career itself. He worked as a postdoctoral fellow at Rutgers University, New Jersey, USA in 1978. He attended specialized course on monoclonal techniques at Royal Postgraduate Medical School, University of London. In his research endeavors, he had research collaboration with many international organizations. He was visiting scientist to Scottish Crop Research Institute, JHI, Dudley, Scotland, Natural Resource Institute, University of Greenwich, Chatham, UK, Hebrew University of Jerusalem, Israel, and Asian Vegetable Research Development Center, that is World Vegetable Center, Taiwan. Getting connected to the institutes across the world located in different continents in the era of no internet, WhatsApp, or any University of California, Dr. Charles K. UK, Professor B.D. Harrison from UK, Dr. Henry Joseph from the University of Jerusalem, Israel, and Dr. Peter Hansen from ABRDC, Taiwan, are the important international scientists with whom he had research collaborations.
Also leaf curl, but the other thing that's particularly memorable is work with the monoclonal antibodies, which enabled us to detect a wide range of Gemuni viruses and to distinguish between them. And the work done on the Indian ones, particularly those from Bangalore, really set up this study in India time. To say how much your contribution to the research is valued, and we hope to keep in touch with you in the future. You'll stay in good health and happiness for many years to come.
VM means only work, no extra talk. He was a very tough teacher, and I saw him as a very passionate researcher. Many things I have learned from him. He greatly influenced me and shaped my professional life. I wish to be very happy and healthy and keep guiding us. Dr. Medinata, we can't have a picture of birthday today. Mariti and I, or particularly I, remember all the generosity of knowledge that you passed on.
Happy birthday, sir. The three words that come to my mind for Dr. Muniyappa are integrity, passion, and commitment. We wish him many, many years of active life and lots of happiness to come.
You have given to us, to many of your students, some of the kind of, the discipline not only helped us shape our career, all the advice and help and kindness of the me. So thank you, and I wish you a very happy birthday this year.
Happy birthday, Dr. Muniappa. 3 words that describe Dr. Muniappa are a completely obsessed scientist. Big memories of Dr. Muniappa with spending time with him are listening to him talking about plant viruses and his concern for everything that other people are also up to. So thank you so much, Dr. Muniappa. Hope it's a great year and lots more years to come. The outcomes from this collaborative research work were published in highly reputed international journals such as Annals of Applied Biology, International Journal of Phytopathology, Archives of Phytopathology and Plant Protection, International Journal of Applied Plant Pathology, International Journal of Pest Management, which was cited by scientists across the world. In recognition his contribution to the plant virology research, he was honored with many prestigious awards and made him occupy prestigious positions. To mention few are, he was an executive member of International Committee on Plant Virus Epidemiological Group, recipient of Jawaharlal Nehru Award in the field of plant pathology during 1975. A scroll of honor conferred for his service by Indian Phytopathological Society in 2002. Fellow of Indian Phytopathological Society, New Delhi. Fellow of Indian Science Congress Association, Kolkata. He was in-service fellow in plant biology at ICRISAT, Patancheru, India, 1983. In addition, he was honored with many awards from different organizations. Behind every successful man, there lies a sacrifice made by their loved ones. As a famous proverb says, behind every successful man, there is a woman. Dr. Muniappa's is not contrary to this. If we don't mention the role of his life partner, Srimati Sudha, and the sacrifice made by her taking the responsibility of homemaker and looking after their kids, we are doing injustice, which indirectly helped Professor Muniappa immensely to pursue his research endeavors. He is blessed with a beautiful family with son Vinay, daughter Shruti, daughter-in-law, and granddaughter, and having a wonderful retirement. However, his wheel of learning never stopped anyway. At the end, it is off to quote Mahatma Gandhi saying today, "My life is my message," which perfectly suits to Professor Muniappa, a sage in research endeavors of plant pathology. The way he conducted himself with a committed pursuance of research career and being humble with great success inspired many young researchers to emulate him. Definitely, his presence today will inspire many more youngsters of current generation sitting here. Professor Muniappa, a sage in scientific pursuits and an unsung hero of UAS Bangalore.
This documentary took us on a brief exploration on the life of Dr. V. Muniappa and some of his inspiring works. I would like to thank Plant Pathology Club for their efforts behind this. Now, I would like to invite Dr. V. Muniappa, Dr. K. N. Ganeshaya, and the Vice Chancellor, Dr. S. V. Suresha, to join us on the dais.
As we gather here to honor the illustrious Dr. V. Muniappa, we'd like to express our gratitude to him by bestowing a special recognition on behalf of our department. I'd like to request our Honorable Vice Chancellor, Dr. S.V. Suresha, and Dr. K.N. Ganeshaya to extend our heartfelt appreciation to Dr. V. Muniappa for his outstanding contributions. I'd also request to I'd also like to call upon Mrs. Muniappa to accept our token of love. Please, ma'am. I'd like to request them to hand over a special memento reflecting Dr. Muniappa's passion to the field of plant pathology. This memento illustrates the interaction between tomato, Gemini virus, and white flies.
Thank you all for honoring someone who has been a role model to many. His works in plant virology has truly inspired us, and we would be delighted to hear a few words from him. Please join me in welcoming Dr. V. Muniappa to share their insights with us.
ಎಲ್ಲರಿಗೂ ನಮಸ್ಕಾರ. Vice Chancellor Dr. Suresh, Dr. Ganeshaya, Professor, Dr. Nagaraj, and ladies and gentlemen. My journey started when I came to Bangalore from Kolar to do my BSc in Agriculture Sciences in 1964, when the college was affiliated to Mysore University, and it was the same year that Dr. Zakir Hussain, the then Vice President of India, inaugurated EAS GKVK, and therefore I graduated from Agriculture University. And that was 59 years ago, and I actively worked until COVID, which was 4 years ago. My work and research took me across the world many times over. I have been guided and I have been inspired by many people. I had the great opportunity to work under late Professor H.C. Govindu. Those three are picture. I had the great opportunity to work under late Professor H.C. Govindu, who was the head of the Department of Plant Pathology, and that is where I learned mycology and inoculation techniques. I did my PhD under Dean, late Dr. Ramakrishnan, my PhD guide. I worked on a lot of disease of rice, for which I got Jawaharlal Nehru Award. I did my postdoctorate on culturing of mycoplasmas in Rutgers University, New Jersey, in 1978, under late Professor Karl Marmarash, who was one of the pioneers in plant pathology, who deserved a Nobel Prize, and he got close to it. I collaborated with Professor B.D. Harrison in the late 80s from Scottish Crop Research Institute, where we characterized 7 Gemini viruses in agriculture and horticulture crops. Today, he is 94 years old, and we still exchange greeting cards and Christmas. And for 30 years, his cards have always been a picture with a bird. During the 90s, I collaborated with Professor John Colvin from Greenwich University in UK, and we have developed tomato varieties resistant to tomato leaf curl virus. This was also the beginning of internet across the world. We were early adopters to internet. I vividly remember going to Videsh Samachar Nigam Limited with the Professor Colvin and my children to apply to obtain an internet connection. In those early days, we needed an explicit permission from the government to have an internet connection. Hence, we at a Bond laboratory became the first users of internet and email, and the university around mid-90s. Things have rapidly changed today. When I was writing scientific papers, I had to type the papers myself in a typewriter that I owned. I often went to typewriter house, Mr. Goindappa, who spent hours typing for me, and then it took days to send out these papers to be published. I had to take the pictures on my camera and get them developed. This had become a routine and used to be my only outing sometimes. The scenario is very different for you students present here today. And you should make the most of technology and benefit from ease of the application on your research and work. I would also urge all of you to publish. My students present here will tell you all. I often told each one of them, publish or perish. And I would say this forever, publish or perish. All of you appreciate
the role of inoculation of virus by whiteflies to seedlings. To do this, I built a vector whitefly glasshouse. At that time, we were working on several Gemini viruses, and therefore, to screen large germplasm, we need a vector glasshouse. Therefore, it is important to build an environment for research. Sometimes they are not readily available, but we have to create one to excel. And in addition to tomato leaf curl Gemini virus, I worked on several other diseases like sand spike, rice elodor, pigeon pea pylori, Niger pylori, and several mycoplasmatices. And Gemini viruses, we worked on, myself, my students and myself worked on Arscramella mosaic, Dolichosella mosaic, tobacco leaf curl virus, tomato leaf curl virus, and so many of them. And other viruses like peanut stripe virus, tomato mosaic, and peanut bud necrosis virus, and in addition to tomato leaf curl virus. And tomato leaf curl virus is an epidemic disease and cause huge losses to the farmers. And we have developed resistant varieties, and also we got a number of genotypes which are pure lines from Asian Vegetable Development Research Center, and we tested them in laboratory at Abboud as well in the field, and also the farmers visited the fields, and we selected out of so many of resistant ones, we selected 3 varieties, Nandi, Sankranti, and Weibo. And farmers also came, they also selected these 3 varieties because they are yielding good fruit size, color, and so on and so forth. And these varieties were marketed for the first time in the Agriculture University, GKVK, and nearly 20 private companies purchased these seeds to use in their hybridization program. To fund research and build an infrastructure around it, one needs to continuously look out for research findings. Unlike today, most schemes and funding are open and published, and information is available on the internet. Those days, it was through a network of scientists, and very often communication occurred through international letters. I must have written hundreds, if not thousands, of handwritten letters. And though one such letter from Professor B.D. Harrison from Scottish Corpus Institute, I learned about British Council funding, which felicitate the most modern equipment for our laboratory. On an Indo-Israel bilateral program, Professor Pulkan also visited Israel in this program. I visited Hebrew University to screen tomato lines to tomato leaf curl virus. The fields I visited were at an arm's distance from Gaza. I encountered shootings in the airport on my return at Tel Aviv, where we had to drop our luggage and run out of the airport. Grateful to Professor Josnek for being around. On return to India, I was investigated CBI because with Israel was not too common in those days. I was often told I was a strict professor. There were times when my student
confided the hardships of working with me to my wife. Today, without an exception, all my students are shining and are on top of their chosen paths. They are in the best of the best institutions or companies under the helm of us. My students made me very proud. One will always encounter bureaucracy at the workplace, be it private sector or public sector or a research institute. However, I am grateful for the university, its then directors and vice chancellors for letting me do what I was best at, to concentrate on research. I'm also very grateful to all my colleagues and students. Dr. Ganesha, I thank you for your excellent talk. Vice Chancellor Dr. Suresh, I thank you for felicitation. Dr. Nagraj and team for conceptualizing and making this very memorable event happen. I thank my friends, colleagues, and family present here, including my granddaughter Aditi, who brings a lot of happiness. Finally, I thank my wife Sudha for allowing me to work in the laboratory late into the night, for driving me to the GPO to send out letters and faxes to make international calls, and for hosting multiple scientists and students at home. She managed everything by herself. Thank you all.
Thank you, sir, for your words. Your dedication to scientific research is truly inspiring, and we appreciate your valuable contribution to our understanding of plant health.
Next, I would like to invite Dr. V. Muniappa's granddaughter to share her experiences with him.
All of you have heard the great achievements of my grandfather and his major contributions to the agricultural field. But let me now tell you the importance and the contributions of him in my life. As I have once quoted before, my tata is my super tata, not only from the perspective of a 5-year-old, but still to this day. The amount of energy he has is incredible. Every day at 6:30, he's out walking and listening to the radio. For every week or so, I am the repair person. To describe him and explain how much I love him would mean for you all to be here till tomorrow. So I'll keep it short. My tata is my role model. He has achieved so many great feats, yet he is so humble. Look at him. At first glance, would you think he's met Prince Charles? I am so proud of him, and for many years I would boast to my friends that my tata met Prince Charles, and they would all be awestruck. I would like to travel the world just as he did. He would tell me many of his stories of his extensive travels. At home, we would watch many Kannada serials together. One such show we would watch was Santam Papam, and whenever we would watch it, Ajji would scold us and we were banned from watching the show. Another show he would watch with a lot of interest was Mahabharat. He was so dedicated to the show, he kept a notebook in which he'd write all of his character names. And if we ever tried to speak during the show, he'd shush us all. Well, what I'm trying to say is that he inspires me every day to be a better version of myself. And for all the gifts and goodies, thank you so much. When people ask me whether I love my father or my mother more, I would say none of the above. I love my tata more. I love you to the moon and back, tata, and maybe even twice that distance. Thank you and happy birthday.
Good.
Thank you, Aditi, for sharing your opinion in such a cute manner. Next, I'd like to request our Honorable Vice Chancellor to extend our gratitude to Dr. K.N. Ganeshaya, who has graced us with an exceptional lecture.
Thank you, sir, for honoring a guest speaker. Now, I would like to invite the office bearers of the Plant Pathology Club, the president, Dr. Nagaraju, the secretary, Dr. C.N. Lakshminarayan Reddy, and the treasurer, Dr. Manjunath B., to join us on the stage to honor our Vice Chancellor for supporting us and taking the time to be with us throughout the event.
Thank you, everyone. As we continue to enjoy this remarkable program, I'm honored to invite the esteemed Vice Chancellor of our university, Dr. S.V. Suresha, who has graciously agreed to deliver a presidential speech. Let's welcome him to the stage for this momentous address.
Good afternoon, respected the former professor and head Department of Plant Pathology, an expert in the field of plant virology, Professor Muniappa sir, and a renowned writer, a speaker, an agriculture scientist, plus an extremely good administrator, Dr. Ganeshaya sir, and my most of the teachers, Dr. Jairamaya sir, Dr. Kulkarni, Dr. Virat Math, Dr. Keshamurthy, and many more who are here for today's beautiful program. Ladies and gentlemen, and it's my privilege to be a part of this program and to wish Dr. Muniappa a happy birthday and a beautifully organized program on his 80th birthday probably signifies with a wonderful program of starting a plant pathology club. And at the outset, let me congratulate all the people who have joined together, started this plant pathology club with beautiful objectives of bringing the plant pathologists to a common platform and igniting the minds of research scholars in the Department of Plant Pathology. And once again, I congratulate all the plant pathologists who could able to start this club in the university. And probably it will be a milestone for other departments to follow in near future. And I need not to say anything much on Dr. V. Muniappa after hearing to Dr. Ganeshaya's wonderful presentation of the personality of Dr. Muniappa, plus the achievements of Dr. Muniappa in the field of plant virology, and relating the recent trends of plant virology to the work or activities carried out by Dr. Muniappa in his service. Probably it's an eye-opener for all of us, how history will remember only a person who has contributed back to the society, rather than who is selfish and conduct most of the things which he can do it on his own. So I hope this plant pathology department will remember Dr. V. Muniappa's work forever, and that will be a guiding force for most of the research scholars in the faculty. And I was really wondered when I saw the people from different departments, the plant breeders, entomologists, the soil scientists. Then after hearing to Dr. Ganesha, I could able to understand that he is not just a plant pathologist, but he is a multidisciplinary person, and which we are talking now. Long back, he worked in different disciplines, but now we are talking about the multidisciplinary approach, which he has shown the way, how it should be. A plant pathologist should also be a good breeder, a good entomologist, in order to bring out best out of his work. The university is blessed to have such a wonderful personality with us. And the Department of Plant Pathology rightly organized a program on this occasion. And you have done a wonderful job by presenting his work through a video, document it, and that you can preserve it in the Department of Entomology. And every year, it should be an occasion to celebrate the birthdays or remembering the contributions of farmer people like this. And at the same time, a word of caution to the organizers who have started this plant pathology club. The associations will start functioning overnight. The groups will start functioning overnight, but sustaining the interest of these clubs for a longer period, bringing out best out of these clubs is very important for all of us. Because any organization, associations or club will have 4 phases. The initiation, the people will be really enthusiastic. They join together and they will show a lot of interest. They will come together. Then after initiation, there will be a stage called rise. And it will be a dramatic rise in the functioning of the organizations. Then the challenge starts for all of us, how to stabilize the activities of this club for a longer period. In case if you fail, there is a worst part of any of these organizations, it will start declining. So in case if you can't sustain the rise of these clubs, at least you have to stabilize at the peak and see that it never declines for any other reasons. So kindly make sure the club function effectively, bring the famous personalities from outside, connect to your researchers, see that they are benefited out of the experiences of these great legendary people, make your discipline rich in contributing to the field of agriculture development, and see that the club will also give it back something to the university in terms of, as he said, publish or perish. And with great pain, I am saying this. Now we have a ranking system in the country where nearly 40% of the scorings is for research and about 50% is for publications. And the university visibility is only possible through the publications in a standard journals with high-NAS ratings. Then only the university will also have that visibility, and university can also rise to that level. And very recently, we have a global ranking of the universities, and we are nowhere in that list. And now the another condition for most of the universities is, if you are in the list of 100 top universities in the world, you are eligible to get certain funds, or otherwise you are not eligible to apply for any of these kinds of funding agencies at all. We just look at the level of competition that we are facing, or otherwise university will also lose its image in the country. So kindly, I hope these clubs will help us to get very good publications, a good contribution in their respective field of specializations, and help the university to have that image in the society. So once again, let me congratulate. And also, you have chosen a right speaker, Dr. Ganeshaya. Other than Dr. Ganeshaya, probably we would not have understood the real personality of Dr. V. Muniappa. So let me thank Dr. Ganeshaya for his wonderful presentation. And also at this age, probably you might have understood how energetic Dr. Muniappa is, how disciplined he is, and he has presented very briefly, but effectively whatever that he has done and what is that he is expecting from this university. So I'm also very thankful for giving me an opportunity to honor Dr. Muniappa, though I may not deserve it. I told Dr. Nagaraj, why don't you find some other better person who can felicitate Dr. Muniappa rather than me? But he said, it is only a positional advantage. I know, and it is nothing else. So I thank the organizers for giving me an opportunity to be a part of this wonderful program. And let's keep moving with the kinds of activities through these clubs. Thank you. Thank you.
Thank you, sir, for your words and for gracing us with your presence. As we near the conclusion of this event, I would like to express a heartfelt gratitude to everyone who has contributed to its success. To formally convey our appreciation and bring this gathering to a close, it's my honor to invite Dr. C.N. Lakshminarayana Reddy, Associate Professor at our department, to deliver the vote of thanks, to share his thoughts and gratitude on behalf of all of us.
Yeah. Good afternoon to everyone gathered here. I think I'm standing between lunch and closing of this function, so I'll go into quickly wind up. So it's my privilege to extend vote of thanks on behalf of Plant Pathology Club, as well as University of Agriculture Sciences, Bangalore. At first, I would like to thank our beloved Vice Chancellor, Honorable Vice Chancellor S.P. Suresha, when we proposed to have this program, that's the opening of Plant Pathology Club, and starting a talk series, or you can say honoring a person that to stature of Muniappa. So he agreed and extended all the, I can say, facilities of auditorium and other things. Apart from that, he is with us throughout the program, even with, I can say, his hectic schedule. So thank you, sir. Thank you on behalf of Plant Pathology Club and US Bank. So today's today's person, we need to actually, with his presence, actually this occasion of opening Plant Pathology Club had, I can say, had a wonderful time. So that's Professor Muniappa, and every one of us know his immense contribution. So thank you, Professor, for being with us and gracing this occasion of opening of Plant Pathology Club. So really, we are honored with your presence, and definitely a lot of new generation youngsters inspired out of you with your presence today. So continuation. So Dr. Ganesha, K.N. Ganesha is with us, and gave a wonderful talk covering, you can say, plant viruses, human viruses evolution, as well as actually, yes, he conducted a research to present a few things on Dr. Muniappa visiting the old laboratories and taking photographs. So thank you, sir. Your presence made a difference today. So from this, a lot of, I can say, retired professors across other, from all the departments, like Professor Jairamaya, Professor Viraktamat, Professor Kulkarni. So they have graced this occasion. Thank you. So thank you, sir, on behalf of Plant Pathology Club. So then, there are several professors here. So I thank wholeheartedly from on behalf of Plant Pathology Club for gracing this occasion, making this program wonderful. And the students and faculty of our department and support staff of our department, as well as you can say estate branch, who extended all the support for conducting this program. As well, if I don't mention, you can say the person who has helped us in making a documentary and recording this and streaming this program. Yeah. So Chandra Mohan is working for a final frames. So I thank you, Chandra Mohan. So our esteemed board members, Professor Chandragowda and Harish, who is with us in this program. Thank you, sir. Thank you for both of you for being with us throughout the program. So I thank one and everyone. If I forgotten anyone, it's not intentional. Okay, I thank you one and all for making this program wonderful. And again, I wish you happy birthday, sir. So we are the one who inspired me to, I can say, work on a plant viruses. To mention on personal behalf, I thank you very much. I adore you a lot. So apart from that, my mentor is here. You also, Dr. M.K. Reddy. So I thank him who mentored me in the field of plant virology, and thank you one and all. And we have a lunch at a botanical garden, so you can join for the lunch and photo sessions. Yeah. So anyone and to have so a small photo session will be there after this program. You can join with us for a photogram of so photography with a professor. Group photo. Okay.
As Sir said, we now invite all our guests and the wonderful audience to join us on the stage for a photo session.